So there we go. Okay, so now we are being recorded. And I can now say welcome to everyone who's joined us over Zoom. It's my pleasure to introduce our speaker today, who's Dr. Enoch Nagoma. Dr. Nagoma is an evolutionary geneticist with broad expertise. He's worked with quite an impressive variety of organisms over the course of his career. I was interested to notice as I was going through his CV. He grew up in Malawi, and he earned his undergraduate degree from the University of Malawi. He then earned his master's degree at the University of Oslo in Norway, where he studied genetic diversity and population structure in the African pencil cedar. From there, he went on to earn his PhD in Germany, where he studied life history patterns and the genetics of age-related diseases in a short-lived fish. And then in 2015, he moved to the US. That's already a very impressive breadth of international academic experience. But when he arrived here in the US in 2015, he went to do his postdoc at the University of Missouri, where he focused on the quantitative genetics of nutrition and energy allocation in Drosophila. So really interesting disparate experiences in plants, non-model animals, model animals. And I met Enoch right when he started his postdoc as his advisor. Dr. Elizabeth King is a close collaborator of mine. And so I'm very excited to announce that as of this year, Dr. Nagoma has accepted an assistant professor position right there at the University of Missouri. And today he'll be talking about the lessons he's learned across the various branches of his career that inform his approach to starting his own lab, where his focus, I believe, will be the evolutionary biology and genetics of aging. So without further ado, I will let Enoch tell us his story.
Thank you, Dr. Burke. Okay. I hope you can all see my slides. Okay. Yeah, so thank you so much for that generous introduction. The slides are not advancing. Okay. Oh, okay, there we go. Yeah, so my name is Enoch Nagoma. I am assistant professor in the Division of Biological Sciences here at the University of Missouri in Colombia. And I will be sharing with you just a few of my experiences over the years, probably not dating back to my master's work, but probably just my PhD and now. So I'll be talking on the topic from the lab to the bush and back, a case for ecological genomics of aging. I guess one thing that's been intriguing to me is that we look at aging mostly from the human-centric view. So we only talk about aging with respect to our own species. And what you're looking at are organisms, fruit flies on the left, and Killifishes at the bottom. And the Killifish story comes from this Fritz Lippmann Institute on Aging in Germany, where I did my PhD, and the fruit fly work comes from MU, which is where I am sitting right now. So aging really is almost universal in the tree of life. So almost all organisms show some signs of aging. I just have a small collection of examples here from like these soil organisms, nematodes. You can actually see aging after time, land-dwelling organisms. So these are like mantids or turtles and those. You can see the mark of aging. And we can also see aging in flying animals, including birds, in swimming animals like fish, and even large-bodied animals, including ourselves as human beings. So it's a pretty ubiquitous process. And what's important is that aging actually has fitness implications. So it's one of those life history traits that reduces fitness. So the more we age, the less able we are able to bear or to reproduce. And that's the same for most organisms. For us humans, we are mostly concerned about longevity because aging comes with disease, and that reduces our longevity or how active we are as we grow old. So aging is a very important question to consider. And here is a cartoon or a picture of a number of different species. And the main point that I want to make is that obviously we see this large difference in how fast or in the pace of aging across species in general. Even if we think of aging mostly for the human race or for the human population, but it's also a species thing. Not only across species, even when we look in one species, I just have one example here, which is a freshwater pure mussel. Across distance in different environmental localities, we tend to see differences in the rate of aging, and not only within across space, but even between sexes, we can have differences in aging. So this is a freshwater copepod where males live something like 5 or 5.5 times less than females. Intriguingly, we can also see aging even within a single genotype, and this is common for eusocial insects, including ants, bees, and others. So even if we think about aging as a human topic, it's really like a species topic. The next thing that I wanted to just share is that normal life history corresponds quite a lot with the rate of aging. So most life history traits, they tend to correlate with the rate of aging. I just have one example here, which is looking at gestation period in mammals. And what you have on the y-axis, that's the rate of aging. And you can see that inverse relationship with between the rate of aging and the length of the gestation period. Just for the sake of curiosity, I labeled that green. That's where I think animals like mice that we use in the lab, for instance, that have a gestation period of just about 20 days, whose log might be around 1.2, and like human beings, where our gestation might be somewhere around 275 days, corresponding to about 2.4 in log terms. You can see how far apart we are. But then these are the organisms that we use to study in order to describe the processes of aging, even in larger bodied organisms. One more thing that I wanted to touch on briefly is that aging is quite related to reproduction. So that's the data that's on the right, where we can actually see, so there is fecundity on the y-axis and mortality on the x-axis. You can actually see that for most organisms, whether we look at birds or mammals, reproductive bout comes somewhere in midlife, but there is a correlation between those two. So we cannot just talk about aging without talking about a corresponding process or a correlated process, which is reproduction. Maybe the last thing, the last general thing that I would like to share is that the rate of aging actually corresponds to the slope of the mortality rate with aging. So this is a diagram of mortality on the left, on the vertical axis, and age on the horizontal axis. And once we hit reproductive maturity, aging begins. So you have that steady rise in mortality in general. And this slope actually is what is different between different organisms or different populations of the same organism. So what I am interested in is what is the mechanism that underlies this variation in slope between populations or different organisms. I just threw in a small tangent probably to help me make the point. So I think that biology is a bit messy. And this is what we know, the scientific method of discovery, where we look at something and when we look at a phenomenon happening in front of us, often that is context dependent. In other words, if I'm looking at a squirrel that's eating something, it might be cloudy, it might be sunny, or it might be raining, or it might be dry. So there is a context that's related to a phenomenon that we are observing. And usually, whatever we make out of that observation tends to be discipline dependent. So a historian and a biologist looking at the same process will come up with two different questions of what is going on. And then that is also based on discipline-specific theory. So the questions that we ask are really, for most disciplines, the theory can be quite rigid. And we do tend to explore those questions using discipline-based theory that usually encourages simplicity or being parsimonious. And then we come to the lab, we try to study the process, it's control and control and control and control, so that we are able to explain what we are measuring. And then finally, we give a conclusion. A conclusion also has to conform with simplicity, and then we are happy. But now the question is, I know that this is the method that we have used and we use, and it is extremely beneficial to what we have known until now. But I guess biology is a bit more messy. The processes are a bit more messy. What I want to dwell on mostly is to present the topic of aging, which is studied mostly as a human problem. I want to present it as a species problem. So it's not just us humans, but other species as well. And there is a whole set of literature out there that sort of alludes to that. And I just put together a few articles that I read, and just highlighting a few words in the titles of those articles, you can actually see that aging and climate change actually might be converging somewhere. So it is widely expected that aging might actually affect genetic diversity of populations, particularly reducing genetic diversity of populations, and therefore affect the course of evolution. So it is a species problem, it is a conservation problem. And my proposition is to begin looking at aging for species as a conservation problem. The key question that I'm interested in is the sort of mechanisms, biological mechanisms that allow the variation in the rate of aging in the wild. I guess that last piece is more important because I know that we know quite a lot that we have done in the lab, but I would like to focus on doing that in the wild. So briefly, I'll be sharing a bit about my experience with genomics in lab adapted inbred fish. That's the killifish that you see there on the right, and some of my experiences with lab adapted outbred populations of fruit flies, as well as how those 2 points are guiding me into where I should go in the future. Okay, so to start with the killifish problem, these are really interesting organisms. They are annual organisms, and so far the shortest lived vertebrate animals that I know. I know that there is a chameleon in Madagascar, but it lives slightly longer than the shortest lived killifish. So normally they live in these really shallow pools of water, even in settled areas, and they grow rapidly, and they will lay eggs. Those eggs will be sitting in the soil, sometimes for years. And then when the rains come, the fish will hatch and carry on its life cycle really fast. So the key here is those eggs going into a process known as diapause or developmental arrest, where they're just sitting in the soil. You can actually bounce it as a ball. It's so hard, and that's how they are. Just to give you a picture, so this is what we did a couple of years ago, where we put data loggers in the wild and just try to study some features of those vernal pools. And so this is a period of 10 months on the x-axis, and we're measuring temperature. And you will notice here that the only time that the water is available is this from between this arrow and that arrow. For much of that 10 months, these eggs are just sitting in the soil in a process of diapause. When the rains come, they hatch and therefore develop very fast and grow and mate, and the process continues. And this is the only vertebrate so far that we know that behaves like this. And this is important for the study of aging in general, because it's a vertebrate, therefore closer to us human beings. We can look at genes that are responsible for aging and extrapolate or try to use those as hypotheses for human aging. Killifish, African killifish are distributed widely in Africa. So all the way, so that green part, all the way down to northern South Africa, much of Africa, you find those fish. So there are more than 60 species of killifish. And the ones that I'll be talking about, these are mostly from Mozambique, Zimbabwe, and there are a few in Malawi. So that's Malawi, that's my home country, that's just above there. And I'll be talking mostly about those two species, Northobranchius cadletsi and Northobranchius fusari. And this fusari is the shortest lived, and that one is a closely related species that's much longer lived. And what's interesting here is that the aridity of the habitat where they are found actually correlates with how long they live, their lifespan. So this lives much longer because it comes from a much wetter area. This is a very short-living species. And that phenomenon also happens within species where we look at strains of just fusari. We can also see significant differences in lifespan and the rate of aging. An interesting thing with this fish is that you can actually see the phenotype of aging on them. These are young on top, the top panel, and these are old in the middle. You can actually see tissue homeostasis failure at the tail. If you compare that with that, that bent skeletal structure, which is what happens in us human beings as well when we grow old, we can see things like loss of color, like bleaching out, as well as muscle loss, that is sarcopenia in human beings. So this visible and vivid aging phenotype is what has attracted aging researchers to look at this species. One more important thing that I would like to share with you is the habitat where these fish live. So as I'm talking about this, think about the kind of environments that we give these organisms when we bring them to the lab. So this is a pool. Look at how dirty it is. And this water was actually collected from a pool where Northobranchius had been caught. Often in those pools, you see animals like livestock like this, or it could be elephants trampling in those pools. And we have this very different just habitat properties. For instance, that's an open pool that's completely covered with vegetation in general. So this is the nature of the habitat for killifish. Yet when we bring them in the lab, we keep them in very clean water with reverse osmosis, with UV devices, and then we study aging on them. Much of the data that I will show is actually coming out of two crosses that I did using the short-lived Nothobranchus fusari, which is on the left here. So I used a male in both crosses for logistical reasons. And I used a female Nothobranchus cadlesi to create those two crosses. And I phenotyped the F1 as well as the F2 fish for both crosses. I'll be reporting mostly on one cross, which is here. And the phenotypes that I measured out of these were actually two that are relevant for this talk, which is lipofuscin. And lipofuscin, this is a physiological marker of aging in human beings. Excuse me. In human beings, we call it the age pigment. It's a product of mitochondria dying and just accumulating, being cross-linked and accumulating in the cells. So it's something that's known from the smallest of organisms like nematodes. So that green patch there, you can actually see the white stuff, that's lipofuscin in nematodes that are 16 days old, as well as in human beings. And the second trait that I measured was apoptosis, which is just cell death. And there is a lot of literature to suggest that apoptosis also is a marker of aging. It increases, the rate of apoptosis increases with aging. And both of these were measured in the liver of Nothobranchus. Okay, so the exact phenotypes that I'm measuring is apoptotic cell fraction, which is just collect the livers, preserve them in PFA, make sections, and stain the sections using the tunnel assay, and then look at the number of nuclei in the field, and or take a fraction of number of apoptotic cells, which are read to the number of nuclei. And a similar procedure for lipofuscin, where I measured the amount of lipofuscin in images and compare that with the number of pixels in the images. So this is a picture of lipofuscin. That's what it looks like. You can't see the cells here because this is just like fluorescent microscopy that was used, but the idea is just to quantify the area of the orange pigments against the area, the number of pixels or covered by the darker background. And for apoptosis, as I mentioned, so the red points, those are cells that are dying or the DNA is breaking. So looking at the phenotypic data, this is lipofuscin, and on the y-axis, that is the deposition of lipofuscin at different ages in Nothobranchus cadlesi, which is the longer-lived species in this case. And we can actually see this increase, gradual increase in the deposition of lipofuscin in general. And the same can also be seen in the short-lived Nothobranchus fusari at 11 weeks and 16 weeks, because this one lives much, much shorter, just about 18 to 20 weeks old, an entire lifespan. And because this was a QTL, or it was a quantitative genetic study, at some point I had to stop the process and use those animals before they die for mapping. So I chose 16 weeks of age as a cutoff where I don't have as many fish that have died for me to be left with a good number of organisms to use. So at 16 weeks, looking at the short-lived and the long-lived, we see a significant difference, which is an important result to indicate that we can be able to map whatever genetic loci that are responsible for these phenotypes. And on the right, there is something interesting happening in that. So the green bar on the left reflects the short-lived, and the red bar on the right reflects the long-lived. And this is the data that I have explained here. But in the middle, the blue bar, that reflects the F1 fish, and the brown bar reflects the F2 fish. And as we can see in the F1 fish, it does appear in that graph that the F1 fish seem to be more resistant in the accumulation of lipofuscin. This was unexpected. And I will come back to that point. And looking at apoptotic cells, something was different compared to lipofuscin. So these are just the same as the graphs that I've shown you before in terms of what's on the X and what's on the Y. And you notice that apoptotic cells, they do accumulate up to a certain age, and then there is a decline towards the end of a lifespan in the long-lived. And that also does happen in the short-lived. So that was one of the differences that we saw in those two phenotypes in those. And looking at 16 weeks, there is a significant difference as well. But looking at the hybrids, it appears to be like an additive sort of distribution of the rate of apoptosis. So this was interesting. It's like probably there are different mechanisms that are underlying these processes. What was intriguing was that when I looked at lifespan, then I saw the same thing being reflected in this fish in that, so that's the short-lived on the left, and the orange one, that's the long-lived species, and the blue bar that is way outside, that's the F1 fish, and this is the same fish where we saw that they were lipofuscin resistant. And the red bar, those are the F2 fish. And the reason why these two lines don't come to completion is because I had to collect those fish for mapping reasons. So that was interesting. At this point, I knew that I'm dealing with something that looks bizarre at best. And then I looked at the genotypic data. So these are just genotypes. It's a simple graph, and I know that it's a very bad graph because of the mixture of green and red, but I'll explain. So this is on the y-axis. Those are the number of individuals that I worked with, and on the x-axis, that's just the genetic difference, the genetic distance across the genome. And the red actually refers where you have red streaks. Those are actually markers that seem to be getting most of their alleles from the male Nothobranchus fusari allele. So in other words, even if phenotypically it appears that we don't have sex ratio distortions, genotypically there were serious sex ratio distortions in general. This is one of those things that are never really looked at in many studies. And I did try to map distortion QTL, which is just trying to find locations across the genome that could be responsible for this distortion of the sex ratio in the genotypic data. And that's what I'm showing there. On top, those are the QTL. You can actually notice you have the linkage groups or the genetic distance on the x-axis and the p-values on the y-axis. There are a lot of those high rises that are actually showing that there are a number of these loci that are active in this population. And those are just genotype frequencies of the same, and you can actually see that red, either those are alleles coming from the male parent or black coming from the female parent. So the point that I want to make is that so these are the p-values that don't conform to the 1 to 2 to 1 ratio, expected ratio of Mendelian inheritance. But again, looking at the genotyping error rate was very low to explain why I have this amount of distortion rate in this fish. And I can only speculate that maybe because it's an interspecific cross, maybe that might be one of the reasons. But it could also be an artifact of lab breeding. And that's where I am going, because I know that with this shortest lived, so the male parent, collected in 1968 from Zimbabwe by hobbies. In 1973, there was a serious bottleneck. Only one pair was left. So everything else that we have right now originated from a single pair from that bottleneck. So there could be something funky actually happening with that. One interesting thing is that you can use that data to sort of MapQTL that are responsible for lipofuscin or apoptotic cells. And here I'm just showing you a linkage map that I constructed. At that point, we did not have the sequence for Northobranchias. Right now, we do have actually three sequences right now, and quite a rich source of genetic tools. But at that point, we didn't have. So what you do is you construct a linkage map, and the points that have been marked there red is those markers that are being distorted towards the female parent, and green those that are being distorted towards the male parent. So you can see quite a spread of those across. And those are the linkage groups where I actually had QTL for apoptotic cells. So QTL, those are just regions of the genome which are correlating with phenotypic data. And those are places where the genes or the polymorphisms that are responsible for this could lie. And two only for lipofuscin. I will not go in details with this because the point that I'm making is slightly different from the data, from the point that I was making at that point. So here is to show you just the effects of the two QTL or the two loci that I get for lipofuscin. I had two genes or two markers that came up, IP07 and GET4. What's interesting in those is that when you look at the x-axis, the left, those are alleles coming from the male parent. The right, those are alleles coming from the female parent. And the middle, those are the hybrids or the heterozygotes. And you can actually see that at one locus, the heterozygotes seem to be fitter or more resistant to lipofuscin, just like we saw in phenotypic data. But for another locus, we see things very different where we have an additive form of inheritance. So that's interesting. When I look at, this is for apoptotic cells. So my title is misleading there. That's for apoptotic cells. The 6 QTL that I got in there, 3 of them actually are showing that the heterozygotes are much more prone, or they are accumulating apoptotic cells at a much faster rate than either of their parents, and that's not expected. And similarly, the other 3 loci, they are showing that the heterozygotes seem to be much fitter, at least, or you might say, roughly additive compared to the parental genotypes. So at best, these results are mixed. It's not what I would have expected. Now, the point that I'm trying to make is that we do have costs by studying laboratory populations, because there are things that do happen in the lab that normally we don't report. So like the fact that there was a bottleneck in 1973, this is something that you don't find in literature per se, except in the hobbyist literature. That's where I read that from. So at best, there is a complex phenotypic and genotypic pattern of aging in this fish. Okay. So I'll cross over to my experience with lab-adapted outbred fruit flies. And this is the work that I have done here, and it's based on a simple principle, which is organisms need food to power their reproductive as well as longevity needs. So here's a squirrel, it's eating whatever it is eating. And you can imagine that if you have a whole lot of nutrients in the environment, obviously there will be more of that nutrient that's allocated towards process of reproduction and growth. And probably, and that actually tends to reduce lifespan in general. Whereas if you have a limiting nutrient source or environment, organisms also have to sort of like decide in quotes or how to allocate that resource, and often they tend to allocate more of that nutritional resource towards anti-aging processes, and that tends to increase lifespan at the cost of reproduction in general. So this is the basic principle on which I am working. And the most or the dominant hypothesis about what genetic mechanisms underlie how organisms partition resources towards either reproduction or somatic maintenance are actually these three hypotheses, three different like nutrient sensing pathways, which is the target of rapamycin, that's the TOR, the sitoin deacetylases, as well as the insulin signaling pathways. So these are the three dominant hypotheses that mediates the rate of aging per se. So for instance, let's take the most common, which is like the insulin signaling pathway. We know that repression of the insulin signaling pathway actually increases lifespan by affecting several processes downstream. So what I have done with fruit flies is to try and use a multiparental population resource that's known as the Drosophila synthetic population resource that Dr. Bark was also, I guess, involved with. I'm not so sure about that, but she knows the resource very well. And the resource was created out of inbred lines, so 8 inbred lines that are collected from everywhere in the world and mixed together for about 50 generations, and that is just like to create a mosaic of the genetic composition at each locus for each of those 8 founder parents, and then inbred for a further 25 generations to create recombinant inbred lines. So those 800 recombinant inbred lines were the source that I started working with. And together with a grad student in the lab, Patricia Williams, and Annika, at that time, she was taking her gap year with us in the lab. We embarked on this project to look for the sources of natural diversity in this population in response to nutritional resources. And what we did was to take those 800 plus recombinant inbred lines, just breed them together, create one large like heterogeneous population or outbred population, which we call the best population, and then take some of those flies, put them on high sugar diet, some on control diet, and some on poorer diet or dietary restricted. So those 3 treatments. And then we collect the heads, the ovaries, and the whole bodies. We sequence those for RNA with the aim of trying to look for whichever genomic loci would be responsible for nutritional allocation. A quick mention about the diets that I'm using here. So I mentioned dietary restriction. So DR, that's dietary restriction, C is control, and HS is high sugar. These are have everything that fly diets have. They have only 2 differences. And compared to the control diet, the dietary restriction diet only has one difference, and that is it has 50% less protein source. Whereas the high sugar, compared to the control diet, has only one difference in that it has about 7 times as much carb compared to the control diet. So those are the only differences that we have in those 3 diets. And let's go. We looked at differential gene expression. So we kept these flies for about 10 days on these different dietary treatments. And all the data that I will show for this will look like this. And this is a volcano plot. And I just want to explain what this means. So on the x-axis, what we have there is just the magnitude of change. So you can, for each of the diet, like high sugar compared to a control diet. So for each of the diet relative to the other, that's the magnitude of expression. And the outer you go, that's the larger the value of expression. And on the y-axis, and that's just the significance for that. And I'm particularly looking for genes that are located within those triangles outside the gray lines. And based on that, here's the data. I'm seeing a large number of genes that are responding to nutritional treatment in both the bodies, whether you're talking of DR versus C, HS versus C, or DR versus HS. Let's look at the heads. It's the same. So it's the genes that are outside those lines and above the gray line. And let's look at the third, the ovaries. It's the same. In total, 2,478 genes are differentially expressed, and 978 for the interaction. This is a huge number of genes. And then what we did next was to look at gene co-expression, and that is just like trying to say, out of these 2,000 plus genes, which genes are being transcribed in response to the same condition of treatment that we're giving these flies. And we can have a scenario where if we have expression on the y-axis and condition on the x-axis, we can actually say that probably for those two tissues, probably there isn't much that's happening across conditions. Whereas in the other scenario, there is a clear difference. You can actually see that probably the B condition is sort of like associated with higher expression in general. So these are the sort of things that I'm looking for in this data. And here is to show the data in the first pattern where we see almost no effects. And these are all significant. Most of them are significant. And in the second pattern where the DR seems to be associated with, or there seems to be lesser expression, at least for some of the tissues. And another pattern where the DR seems to have higher expression, differential expression for some of the tissues. And another for where both the DR and the HS seem to have like higher expression in some of the tissues. So there is a wide, I got about 22 of these modules or gene co-expression modules, and it was hard to pinpoint which one is more important than the other. So the last thing that I did with that data was to look what is known as gene set enrichment analysis. That's just like looking at sets of genes that have been already predicted to statistically correlate with whatever I'm looking at. So here's the data. I find a large number of GO terms. So in this case, they are GO terms, but they are also pathways. I find 4 pathways, which are listed at the bottom in a blue bar, and a large number of GO terms, 41 in total, that represent signaling, metabolism, morphogenesis, and various other things. So I can't narrow that large gene set, gene data into a few processes that could be important. So this was really interesting in the sense that what is missing might be more important than what is available. So what is missing here? There are those 3 pathways that I have mentioned before. The insulin signaling pathway, it's not there. The TOR pathway, it's not there. The sirtuins are not there. So this is really interesting. So are we really measuring the things that we are intending to measure, or are the mechanisms that operate in bred lines in the lab the same that operate in outbred lines or outbred populations in the wild? Probably not. To supplement that data, what I did was to quickly look at just the insulin signaling pathway take 260 lines, cross them to one of their founder parents and put those on different diets and look at expression of insulin signaling genes. What do we see? About 45 of the 54 genes that we assayed actually were significantly differentially expressed. But look at the size of expression, of differential expression, very minimal. So these results put together actually suggest that probably there are more processes that are happening if a population is outbred or if a population is natural than if a population is just like a lab population that's a lab adapted and population. So there is a complex mechanistic pattern of aging in general. And based on this, what I am now currently doing is to try and bridge this gap, to try and look at the field using both organisms, the fruit fly and the Northobranchias. In this case, you can actually see that Northobranchias has a fruit fly lifespan, but a vertebrate genome, which is really important. And we can actually do good studies across the two species. And to do that, I want to follow an integrative approach where I am not just looking at the genome or the transcriptome, but I'm going through what's known as the endophenotype, that is the other levels of organization to study the rate of aging. And looking at more time points, so going out to the field and looking at different time points, I'm proposing about 3 years, look at what is happening in the field for 3 years, look across organisms, either killifish or fruit flies, and look across environments. So we know that the more arid environment, the lesser or the lower the lifespan, or the more rapid is the aging. And then try to combine fieldwork, wet lab, as well as computation, and involve students in that kind of work. And in order to do that, I have aligned 2 main projects. And let me say that I'm hiring grad students. So if any grad students are interested to look at killifish or fruit flies or anything in between, let them contact me. So mechanisms of aging rate in killifish to identify critical environmental cues. So we don't know what changes in the environment actually translate into the change in the aging rate. And identify target genes, gene networks, or what is known as network adaptation in general, in the wild, in different localities where we can identify a dry and wet contrasting environments. Okay, maybe I will skip this. So this is just like the details of things that I would like to do with that. I may just skip that. The second project is actually to look at fruit flies. I also would like to use fruit flies that have been collected from the wild as well. And in that case, to localize target gene networks. So the idea is if I can have a bunch of loci that are responding to these in fruit flies, are they the same loci that we identify in killifish, in wild organisms? Are those the same loci that we identify in laboratory bred organisms, adapted organisms? Are they the same or they are different? If different, what are those differences? So to do that, I will do an experimental evolution study that depends on just withdrawals of flies at certain points for a great number of generations and sequencing that data. So we can talk about that if some are interested. So just to sum up, this is like this is like the scheme that I would like to look at things through, where we are considering several environments, or at least contrasting environments, using a longitudinal sort of like paradigm, longitudinal studies, but also paying attention to various levels of biological organization and trying to compare two organisms and try to integrate that data. What are we going to get? What are we going to find? So this is where I am going. And lastly, just to thank a few or a large number of people actually, and collaborators that have stood with me over the years. So my PhD lab, my advisor was Alessandro Cirellino in Germany, and these other groups at the Fritz Lippmann Institute. Here, Dr. Elizabeth King, and a whole host of students that have been in the lab, undergrads, as well as graduate students, and collaborators, Martin Reichert, who does ecology of killifish in the Czech Republic, and the labs in Germany that I collaborated with, and also the funding sources. I am happy to take questions if there are any. I know that has gone up to, that's 452 that side, I guess.
Yeah, thanks, Enoch. I'll try to facilitate questions where I may, but you might also see hands pop up yourself. I'll read. So first of all, that was a wonderful talk. I want to make sure I said that I found it very interesting. And Jamie Cornelius in the chat has asked, she says she really enjoyed your talk as well. And she asks, are there markers of age in the blood that can be used to allow for repeat sampling? We think about metrics of oxidative stress, but we're not aware of lipofuscin.
Yeah, so in the blood, so underline in the blood, I don't know if there are any in the blood, but there is a whole host of markers that are known as, that are grouped in the same basket known as advanced glycation end products that includes a lot of physiological markers. Maybe some of them may be in the blood. I haven't come across a specific blood marker, but there is a whole host of markers that people have used or are using. And probably that might represent an interesting area to try and identify if there are any, which I would doubt that there are none in the blood. Probably there could be. So one thing with this is that it's a very new model. The first paper was published in 2003, and most of the work that has been done actually from 2006 to date. So you can see that this really, there is a lot of ground that still needs to be broken for this, but it's a very important, or it's a very promising model, not just for aging, but if you want to look like speciation, for instance, sex-based traits or things like those. So there is a lot of ecological value as well in this. I'm not sure if that's a satisfying answer for you. Okay.
Thanks, Enoch. Are there other questions? Feel free to, actually, I'm not sure how webinars work exactly, but I think you can raise your hand or simply just type a question into the Q&A for Enoch to answer.
Okay, so there is a question. I enjoyed the talk. As you move across different levels of biological organization, e.g. genome to metabolome, etc., do you have plans to account for differences in evolutionary repeatability across these levels? For instance, metabolomic convergence might not require genomic convergence. That's a fabulous question. Actually, right now, so if you notice where I describe these levels of organization, I think metabolomics and microbiomics, I italicized them, and I put in green the transcriptome, the genome, and the phenotype. Those are like the three levels where I have experience, but metabolomics and microbiomics, those are the two levels where I want to grow into. So you are right. We know that right now I'm doing a very small grant for the Nathan Shock Center, where we are trying to do a pilot study for the metabolism of fruit flies. This will be done at Washington University. And the idea is, we know that at least the metabolism is supposed to, because it's immediately below this the phenotype level, we should be able, so like for our population, we don't see, after 65 generations of selection for fruit flies, we don't see differences in lifespan. We see a ton of differences in terms of gene expression and sequence variation. Probably if we look at the metabolism, we might be able to see how genetic information filters out into the phenotype. We might be able to see that. So that's my future direction. I haven't done that yet.
All right. Well, I think I'll ask a quick question. I don't see any other questions from the audience just yet. So I'll jump in and say, I think it's so interesting that when you try to look for evidence of these genes, which are supposed to be canonical, like insulin signaling TOR, sirtuins, and you don't find evidence of them in these laboratory experiments with outbred populations that are designed to map these traits. So I feel like we're sort of seeing more and more examples of those pop up, especially with regard to the studies of aging and longevity. So I guess the question I have for you is, what do you think of the criticism that these are highly artificial lab populations in the first place? And should we expect the results from those lab experiments to translate to nature, where, in nature, populations may not be as outbreeding, for example?
Yeah, I guess it was my motivation for showing those really dirty water where my fish come from. So I guess, so in the data that I showed, RNA-seq data, for instance, so there are a lot of TOLL, T-O-L-L pathway genes that show up in there as well. So these are genes that mediate immune responses and things like those. I don't know why those genes show up. It's just an area that just has not been looked at. And I do agree with you that, so let's think about food. We give them preserved food in the lab. Nutritional diversity in the lab is too little compared to what they eat in the wild. So these fish, they eat a lot of organisms in the wild. We can't do that in the lab. And then let's talk about lighting. We give them artificial lighting in the lab. They have white light in the field. Temperature is the same. We have constant temperatures in the lab. Temperatures are so variable, even on a daily basis in the field. So there are a lot of things that are happening. And that's why I think that for the sake of studying aging, maybe for the human population, what we're doing right now works. I'm not sure that it works perfectly, but if we are to look at aging as a solution for saving species in general, maybe we need to be emphasizing more on ecological genomics, going out, looking in the wild, sampling in the wild. And I think that now the tools are there in terms of genomics, sequencing and stuff like that to be able to do that in general. So I do agree with you. I don't think that we should expect that things will be one-to-one between the lab and in the environment. I think in the chat.
Okay. As far as I can tell, the chat messages are just thanking you for your talk, and I don't see any other new questions. I'll maybe just pause for a second to make sure there aren't any additional comments or questions that pop up.
As we are waiting for that, if there are undergrads that are thinking about doing something interesting, looking at aging from an evolutionary perspective, and they want to look at fish or fruit flies, and just get in touch with me.
All right. Well, it's a little after 5 here, and it's even later for you. So I think that with that, we will thank you for your time. We really appreciate you giving our Zoom seminar today. And we'll let you go. Thanks again.
I appreciate the opportunity. Thank you so much.